We're here to talk about genetics of the virus instead of the pig. So 101 uses for PRRS infectious clones. Some of you I know work with infectious clones of PRRS almost every day. Most of you don't. And this talk is targeted for those of you who aren't quite sure what an infectious clone is or how you make it or how you use it. So let's start with some definitions of terms. Genetics, or forward genetics, or classical genetics, is when you start with a mutant phenotype and you try to determine the genetic basis of that mutation. Reverse genetics is the opposite of that. So you're starting with a genetic mutation, often an engineered mutation, and you ask yourself, what phenotypic effect does this have on your organism? And it's not just for RNA viruses. You can do reverse genetics on any organism that you can manipulate the genome in specific molecular ways. RNA and DNA viruses, single cells, bacteria, mammalian cell cultures, plants and animals. And as we just saw, when you knock out sialadhesion or CD163 in a pig and then ask the question, what effect does it have on PRRS replication, you're doing reverse genetics. So how does reverse transcriptase come into the picture? Well, with RNA genomes especially, there's an extra level of difficulty because we don't have the molecular tools to directly manipulate RNA genomes compared to DNA genomes. So the solution is to make a DNA copy of the RNA, or a cDNA. And we do that using an enzyme, reverse transcriptase, which everybody knows comes from retroviruses, also a few other virus families, and from transposons. And the cDNA is easy to manipulate. We can make our modifications and we can transcribe it into RNA, which then represents the PRRS virus genome in this case, using ordinary RNA polymerases. The modified RNA genomes are then introduced into a cell using transfection techniques. And what you get coming out is a modified virus that has your mutation in it, and you can assess the phenotype. Hopefully that's what happens, but often the result is a lethal mutation where you don't have any infectious virus coming out, and that tells you something about your mutation. So it's just a coincidence of words that the reverse genetics for RNA viruses requires the enzyme reverse transcriptase. So how does one make an infectious cDNA clone? I'll go through this quickly. In the upper left, we start with the virus, single-stranded RNA genome in red, isolate the RNA, make a cDNA copy, and clone that copy into a plasmid that contains the promoter. In this case, the promoter is a bacteriophage promoter, a prokaryotic promoter. Relatively short, relatively simple, useful for in vitro transcription. So once we have the genome in the plasmid, we can introduce our mutation, which could be a point mutation or multiple point mutations, an insertion, a deletion, or a combination. And then we have in vitro transcription in a test tube where this promoter, in the presence of the right polymerase, will make an RNA transcript that contains the mutation of interest. That can get transfected into a cell and you get out, hopefully, a mutant virus with your mutant phenotype, and now you can ask the question, what was the effect of that mutation on the virus? This is called an RNA-launched approach. The other kind is DNA-launched. Look for the changes. Two changes. The first one is the promoter. So this is now a eukaryotic promoter, one that works in this cell. We use a CMV promoter. And the other change is that in vitro transcription is missing. So you don't need that step by putting the plasmid directly into the cell. Some of it will get transcribed into RNA, making your modified genome, which will make your modified virus. Not only do you save a step, but this method is about 100 times more efficient at making mutant viruses. This is a short list of some well-known cDNA clones from the literature. There's only 8 here. There's at least 22 in the literature already and more every minute. Apologies if your favorite one isn't on here. This collection is largely historical and also North American centric because of the nature of the meeting. So what use is an infectious cDNA clone? There's over 100 peer-reviewed journal articles talking about how we can use them. So now I'm going to go over each one of them in great detail. Yeah. How about 9 examples? We can do 9, right? So let's start with the P129 clone, which was made in our lab. One of the first things we wanted to do when we had our clone was to say, can we make a simple gene knockout vaccine? Delete one of these genes, structural genes is what we targeted first, to see if we could have a replication-competent virus in a cell line. So we did that. I'm showing here deletions in ORF2 and ORF4. We also did 3, 5, 6, and 7. And our first conclusion was, no, you can't knock out any of those genes and get a viable virus. But we were very careful. We didn't delete the whole gene up here. We deleted the central portion, making sure we didn't interfere with flanking genes, either by deleting an overlap area or by deleting a TRS, a transcription regulatory sequence for the downstream gene. And we looked, and the other genes were all being expressed. The one we deleted was not. So then we said, well, what can we do about that? Can we make a complementing cell line that expresses an intact copy of the partially deleted gene, infect that, and now the deleted virus is getting the gene it needs and trans from the cell? And we were able to do that for 2 of the 6, for ORF2 and ORF4. And seemed like a good idea. We actually went as far as to put it into pigs because we could get a pretty good titer when we grew them on the cell. But maybe not surprising, this is a single cycle vaccine. It only replicates once in the pig. Therefore, it's a totally safe vaccine. It doesn't shed at all. But you only get that one round of replication to give you the antigenic mass to get immunity. And we just couldn't get enough antigenic mass from a single cycle to stimulate robust immunity. Okay. Second example, this is a much simpler one. This is in the VR2332 infectious clone, which is the VR2332 is the parent of the MLV vaccine. And it was noticed that there were some particular amino acid changes between the vaccine and the parent. One of those was phenylalanine at position 10 in ORF2. And that seemed to correlate with the ability of the vaccine virus to grow well to high titer on marked cells, whereas the parent of the vaccine didn't grow as well. And so what KJ Yoon's lab did was to put in that one amino acid change in this infectious clone, and suddenly it grows better to a higher titer and with a large plaque phenotype. So this is a very straightforward example of how a single amino acid change, you can test that to confirm it has the phenotype that you think it might have. This example is with the Lilstad virus infectious clone, the first PRRS infectious clone. Looking here at a portion of ORF1A, NSP1 alpha, NSP1 beta. These 2 genes are very similar to each other, probably a result of gene duplication in the past. They have very similar protease domains. And the question being asked is, do these 2 genes have different functions in the virus? And so that individually they knocked out the same protease domain in either NSP1 alpha or NSP1 beta and got these results. Knockout alpha knocks out subgenomic RNA synthesis only. Knockout NSP1 beta knockout subgenomic RNA synthesis and genomic RNA synthesis. So here's an example where 2 genes with similar functions really don't have the same function, and here's how you show it. This is Yingfang's group in South Dakota. They have 2 infectious clones, more than that actually, but 2 that happen to represent type 1, that's Euro-PRRS. So it's type 1 PRRS isolated in the U.S. And a more conventional type 2 infectious clone. And we're still looking at NSP1 beta up here, but a different function. So a lot of PRRS genes have more than one function. So NSP1 beta is known to inhibit, to antagonize interferon induction, right? So there's a very conserved, totally conserved, in both genotypes, sequence of amino acids in NSP1 beta. And the question was, if you knock that out, you know what happens? And the answer is, if you knock it out in either infectious clone, you eliminate this, you get more expression of interferon alpha, interferon beta, and interferon-stimulated gene 15. In other words, you eliminate that function of the gene, which is to antagonize interferon induction. The next 2 examples I'm going to show have to do with marked vaccines. But just to get our terminology straight, there's 2 kinds of marker vaccines. And it's kind of showed up here along with the conventional ELISA, a DIVA ELISA, and a compliance marker ELISA, and what you'd expect to see from different serological statuses of pigs. So a DIVA marker, that's differentiating infected from vaccinated animals. These markers are made, they're negatively marked. They're made by removing B-cell epitopes from the virus so that your vaccine virus doesn't have a serological response to those epitopes. Whereas field viruses or other vaccines will have a serological response to those epitopes. And so your ELISA is looking for those, and your vaccine, your DIVA vaccine, is invisible to that new ELISA. But that new ELISA would then pick up any other field strains that are in there. And so this is useful when you're trying to keep your herd negative, but you want to use a vaccine. Use a DIVA vaccine, and if you have an incursion of another strain, you can detect it very early if you're monitoring on a regular basis with this other ELISA. Okay. In contrast, the compliance marker is a different thing. This is positively marked. It's adding epitopes, B-cell epitopes, to your vaccine from a non-PRRS, an unrelated, something pigs wouldn't even see. Maybe just a random sequence. And therefore, your vaccinated animals, if they actually got vaccinated, and if they responded to the vaccine, then you get a serological response you can follow, and you can confirm that they actually saw the vaccine downstream, perhaps when those pigs change ownership. So several different groups, and you can see them listed here, all at about the same time made deletions in NSP2 in an attempt to make a working DIVA vaccine. And with some success, too. These mutations, NSP2 is kind of notable in that it tolerates fairly large mutations, unlike essentially all the other genes in the virus. So you can knock out multiple B-cell epitopes. And then other groups did the same thing, but these groups also had an ELISA that would detect field strains and not detect the knocked out virus. So good steps towards a working DIVA vaccine. Here's an example of a compliance marker. This is with the Chinese PRRS infectious clone. And what they did is they got rid of the first 11 amino acids in ORF7, that's nucleocapsid, and replaced it with this sequence, this synthetic sequence, which doesn't mean anything except that there's a monoclonal antibody that reacts with it. So they started with the reagent and went toward the peptide. That gave a viable virus, grew well, and acted just like the parent, essentially. But it left a serological response to the foreign peptide, so you could go into a pig later, and you could tell it had been vaccinated, even if it had also seen field strains in other vaccines. So you can make chimeric viruses and ask various questions about that by swapping in individual ORFs or clusters of ORFs into an infectious clone. This involves both the PRRS Chinese type 2 clone, little Lillistat type 1, and another arteriovirus, equine arteritis virus, EAV. EAV has a broader cell tropism than PRRS. PRRS has a very narrow tropism. So the question was, what structural genes, what envelope genes are responsible for this difference in cell tropism? So one thing they did was to swap in the ectodomains of GP5 and M, that's here, from PRRS into an EAV infectious clone, and it still had the EAV cell tropism. Then they did the reciprocal, which was to swap in these genes from EAV into a PRRS infectious clone, and it still had the PRRS cell tropism. So ORFs 5 and 6 actually were not important in this broad versus narrow cell tropism. But later, there was a swapping in of ORFs 2, 3, and 4 from EAV into PRRS, and you end up with a PRRS chimeric virus with the broader cell tropism that's associated with EAV. And that makes sense now, because unlike the ORF, the GP5M heterodimer, the GP234 heterotrimer is known to bind to CD163. So you can shuffle at the whole gene level, or you can shuffle tiny bits of genes. So XJ Meng at Virginia Tech has been publishing some papers recently using molecular breeding or gene shuffling. That's a way to get a gene with up to 7 parents in this case. So random bits and pieces of these 7 homologous genes were shuffled, and you get out a nearly infinite variety of random genes that are the same length, basically the same overall structure, but the individual amino acids came from one of 7 different parents. Okay. And when that was done individually with ORFs 3, 4, 5, or 6, it worked. Some of them were viable and grew well, and some of those showed a broader cross-neutralization spectrum than the parents did. So there's a good step toward a different way of getting better cross-reaction. And this is my last example. We're back to P129 virus. Can we make a PRRS expression vector? All right. Can we make a general purpose vector for expressing foreign genes within the PRRS genome? We did that actually some time ago. There's a unique restriction site here, MLV1, and another one, AFL2. And so you can do directional cloning of any gene you want in that site. It's located between the non-structural genes and the structural genes. In this case, the TRS that drives the foreign gene is the TRS that would normally drive ORF2, but now it drives the foreign gene. So what drives ORF2? Well, we added an extra copy of the TRS from ORF6 directly in front of 2A and 2B, and that's built right into the vector. So the empty vector replicates fine. It's actually got 2 TRSs now driving ORF2, but that doesn't matter. We can put various things in there. There's probably a size limit. We haven't pushed the size limit. But we have, between these different groups, there's been a lot of genes gone into this vector. GFP, enhanced GFP, red fluorescent protein, Ranilla luciferase, different interferons, alpha, beta, delta, omega, interferon 2 beta, or 1 beta, I'm sorry, and PCV capsid protein, right? So the results are pretty good. We looked at stability in the GFP construct. We could go out at least 37 passages with complete stability, which is an improvement over previous versions that inserted GFP in frame with one of the PRRS genes. Also, antibodies in pigs have been induced. Dong-Wan Yu did this study, induced in pigs to both the GFP and the circovirus construct. So in summary, PRRS infectious cDNA clones are a reverse genetic system for PRRS, and they work pretty good, and we know how to do that now. Many uses for them, gene function studies, cell tropism, adaptation of cell lines, mechanisms of virulence, attenuation in vaccines, increasing immunogenicity in vaccines, trying to figure out how PRRS inhibits or causes immune dysfunction, cross-protection for vaccines, different ways of increasing that, marker vaccines, both DIVA markers and compliance markers, multivalent vaccines, with the circovirus capsid in there, potential bivalent vaccine. And also, I didn't mention this, but you make just green and red viruses that glow. The infected cells will glow on an FA scope. And those can be really handy for a variety of reasons because you can see them growing in real time. You can look at the expansion of the foci and the spread over the plate. You can look inside that cell and see where the GFP is being expressed, where the virus is replicating. We also use the green version of this, which we call the Kermit virus, to look at our cDNA library and pull out CD163 receptor, because we could find 1 in 10 million cells and identify, okay, this pool has one clone that contains whatever the PRRS receptor is, and we can try to fish it out of there. And as we enrich for it, we knew we were enriching for it. We didn't have to do duplicate wells and fix and stain one and hope it was in the other one too. So in conclusion, it's been 18 years now since the first PRRS infectious clone, and we do have good reverse genetic system. The infectious clones have been invaluable in driving advances in PRRS virology and vaccine research.